Hi everyone, welcome back to the next video. We'll continue our discussion on epistasis. So we saw last time that if the contribution made by a particular locus is independent of what is present elsewhere, then movement on that adaptive trajectory leads you to the same final conclusion, same final sequence which has the maximum fitness. In the toy example that we discussed in the last video, that sequence was GCA, irrespective of what the starting sequence was. That is a case when epistasis is absent. So if we look here, more often than not, what is going to be the case is that the fitness contribution, let's imagine again this 3 base sequence, and we could have 2 scenarios. In scenario 1, the fitness contribution of A at position 1 is simply 0.2 and is invariant of, it does not depend on what is present at position 2 and position 3. On the other hand, we have this scenario 2 where fitness contribution of A at position 1 is actually dependent on what are the nucleotides that are present on position 2 and position 3. This phenomena, which is that fitness contribution of a locus is dependent on other loci in the genome. It could be just another one locus or multiple loci in the genome. This phenomena is referred to as epistasis. In the case of no epistasis, fitness contribution of A at position 1 is simply 0.2 and this is independent of what is present at other loci. At other loci, which is positions 2 and 3. And this is a case when there is no epistasis. We also saw in the previous video that in the case of no epistasis, the implication here is that irrespective of starting point, you end up at the highest fitness sequence. Highest fitness sequence is reached. One of the questions of big interest in evolution is how predictable is evolution. And in one of the popular manifestations of this question, a renowned paleontologist and evolutionary biologist Stephen Jay Gould, who is no more now, asked a question in the late 1980s. And the rhetorical question that Gould asks is that the Earth, the planet that we live on, is about 4.5 billion years old. And the rhetorical question that Gould asks is that if we were to go back 4.5 billion years ago and restart the formation of planet and everything that has happened since, if we let Earth evolve once again, would evolution take a similar trajectory that it has already taken, and bipeds like human beings would be the dominant species on the planet, or evolution is a process is extremely noisy, and hence it could end up in any other direction, and it would be very rare that it would be bipeds like us who are the dominant species. And this is also a question of chance and determinism, which is a more important factor that dictates evolutionary trajectories. If we rerun the Earth experiment 100 different times, and every single time it's Homo sapiens that have come to dominate the planet, that means there was some necessity in the evolutionary process itself that species like us would come to dominate the process. However, in these 100 runs, if the evolutionary outcomes are completely different and organisms of different shapes, sizes and morphology are coming up every time, that means evolutionary processes are dictated by chance events to a large extent. What no epistasis does, it says that irrespective of your starting point, you reach the same peak every single time, which makes the whole process of evolution predictable. So that's one consequence of epistasis not being there, that this makes evolution predictable. In the context of fitness landscape, this can be viewed as the follows. Let's imagine that this is the sequence space. Let's imagine that this is the sequence space. So, this two-dimensional space is the sequence space where I am drawing all the nodes, and this third dimension is the fitness. And here I have nodes, and each node is pulled up to an extent which is proportional to its fitness. What lack of epistasis would do, it would give the structure, it would give the fitness landscape a structure like this. A single peak which is a smooth structure. So, let me complete this again. This would be what the structure would look like. What this means, that if I have a starting population which is here in the sequence space, this is the node that the population starts from, its fitness is very low, but it will keep on acquiring mutations and move up this fitness landscape, because every move up is preferred by natural selection and is selected for, and eventually I will end up at this particular fitness level, which is corresponding to a node which is here on the sequence space. However, if I started from another place in the landscape, let's say somewhere here, then natural selection will still force me up this fitness peak, and I will end up at exactly the same position as before, irrespective of where I start with. And I can choose any starting point on this landscape, and the end position would be the same fitness peak, which is the only peak which is present. So, if there is no epistasis, then I have single peak, and any starting point leads me to this peak. As a result of this, evolutionary process in the absence of epistasis is predictable, because I know that it does not matter where I am starting with, the endpoint where this sequence is going to end up is actually already defined. But it is not as simple as this, because epistasis is pervasive. This phenomena of epistasis that we are discussing is just pervasive in biology. So, epistasis is everywhere. That means evolution is dictated by epistasis, and hence is not predictable to the extent it is when there is no epistasis. So, let us see what does epistasis do. To the same structure, if epistasis was present, the fitness peaks may look like this. This is the sequence space and this is fitness. So, now what has been shown in this figure, that there is not one but actually two fitness peaks in this landscape. So, if I start from here, then I am going to move up and I will end up on, let us say, peak 1. If I start from here, I will end up at peak 2, but so there is some degree of unpredictability involved, that depending on where I start on the fitness landscape, the final destination of the peak that I am going to end up in is determined. So, just to label this fully, we have this as sequence space and this as fitness. But even more surprisingly, what happens if I start here? If I start in the valley between two peaks, then if the first mutation takes place in this direction, then I move towards this peak. But if the first mutation that takes place takes me towards this direction, then I move towards this peak. So, what these illustrate, what these two cases illustrate, is that depending on starting position, a population ends up on peak 1 or 2. So, if the starting position is this one, you end up on peak 1. If the starting position is this one, you end up on peak 2. However, this particular case represents another, this represents another case altogether, where your starting position is here, but despite the starting position being here, it is not clear where you will end up. Sometimes you may end up on peak 1 and sometimes on peak 2. So, from the same starting position, the population could end up at peak 1 or 2. Which is really surprising, because now you are saying that it is the first mutation that dictates evolutionary trajectory, first mutation towards peak 2, and then you have started climbing. So, hence you can't come down because natural selection will not permit it, and so on and so forth. So, epistasis, the net result of all of this means that epistasis makes fitness landscapes more rugged. Ruggedness is a concept that's used to define fitness landscapes and is a measure of number of peaks that exist in the landscape. So, this structure is rugged because it has 2 peaks. So, epistasis makes fitness landscapes more rugged, and as a result of that, evolution more unpredictable. And this unpredictability comes from 2 facets. One is that depending on the position that a population is occupying on fitness landscape, you could go to peak 1 or peak 2. That is a function of where one is located on the landscape. And the other unpredictability is that there might be some places where sometimes you will end up on peak 1 and sometimes you might end up on peak 2. So, this is sort of the concept of epistasis, which brings about its own idiosyncrasies while defining evolutionary processes. More formally and mathematically, epistasis is defined as the follows. That imagine a DNA sequence. This DNA sequence has no mutation. This DNA sequence might acquire one mutation. Let us call this mutation A, which brings about a change of delta F1. What that means is that if the fitness of the ancestor was F0 and the fitness of mut1 is F1, then delta F1 is simply F1 minus F0. This could be beneficial, this could be deleterious, we are not making any distinction at this point. Let us also imagine another mutation. This is mut B. And mut B has a fitness F2, that means the effect of that particular mutation delta F2 is just F2 minus F0. And lastly, we have another sequence which is a combination of both these mutations, which is mut B and mut A. Suppose we have a genotype which has both these mutations. Let us call the fitness of that genotype as F12. So, delta F12 is going to be F12 minus F0. That is the scenario that we have. If there was no epistasis and these mutations were making their effects on fitness independent of each other, then you would imagine that delta F12 is simply a sum of delta F1 plus delta F2. That having both these mutations would simply add up the effect of the 2 mutations by themselves singly that they did. But that is the case of no epistasis. Sometimes, so epistasis can play out in the following fashions. Let us imagine a scenario where only mut A happens, mut A. And without any loss of generality, let us assume that mut A and mut B by themselves are both beneficial mutations. Up here is we are saying what is the effect on fitness. So, let us say mut A has this much effect on fitness. This is delta F. So, delta F1 is this much. Similarly, we will do the same for mut B, which is also a beneficial mutation in the case we are discussing, and let us say its benefit is slightly bigger than delta F1 and this number is delta F2. That is mut A plus mut B. Now, that is these two, and these two in these genotypes are the only mutations that are present, but now we will try to pose the different possibilities of what might happen in a case like this when both the mutations are present. So, when mut A and mut B are both present, then one of several things could happen. The most simplest case that could happen is that the effect is simply addition of the two effects. This is the simplest thing that can happen. This is just delta F1 plus delta F2. This is a case of no epistasis. On the other hand, you could also have a case where the effect of the two mutations is greater than the sum of the two individual mutations effects. You could have this scenario play out. In this case, delta F12 is bigger than delta F1. Let me get rid of this. Delta F1 plus delta F2. So, the actual effect of, in the latest case that we have drawn, the actual effect of having both the mutations is greater than the sum of the two mutations that we started with. This scenario is referred to as positive epistasis. In another scenario, the effect of these two mutations could be something like this, that on the whole having these two mutations confers a benefit which is beneficial and bigger than delta F2, but it is less than the sum of the benefits that we observed in the no epistasis case. In such a case, epistasis is said to be negative epistasis. In this case, delta F12 is less than delta F1 plus delta F2. That the net benefit of having both the mutations present in the genome is less than the sum of the two the benefit conferred the sum of the two benefits conferred by these mutations when they are present singly in a genotype. And the final case of epistasis that we will look at here is that you can also have cases where something like this might happen. This tells you that even though having mutation 1 or mutation A was beneficial because it increased fitness, having mutation B was also beneficial, it also led to increase of fitness, but having mutation A and B and mutation B in the same genotype was actually deleterious. For some reason, having both these mutations led to something led to the detriment of the cell, and as a result of this, the individual that was carrying both these mutations was less fit as compared to individuals that was only carrying either one of the two mutations or even the ancestral one. Having both is a deleterious effect on fitness. This particular manifestation is known as sign epistasis. And there are more intricacies and complications in each one of these cases. But by and large, these are the three major divisions that we will be interested in when we are discussing epistasis. That in the case when there is no epistasis, the fitness effects in the double mutant are just sum of the two individual mutations effects. In positive epistasis, double mutant has an even greater benefit than the simple sum of the two individual mutations. In negative epistasis, benefit is less than the sum, but it is still the benefit. And in sign epistasis, the nature of the mutation flips as compared to the two individual ones where they were found to be beneficial, and now combined together, it actually turns out that this is a deleterious combination. As a result, in sign epistasis, this individual is of a much lesser fitness. So, the net result of this discussion being that epistasis is a ubiquitous phenomena in biology. It dictates evolution, it dictates cellular function in ways that we are still just about beginning to understand. And from the context of fitness landscapes, it makes these landscapes multi-peaked, as we saw. And in this multi-peaked structures, because of the structure that we have come up with, evolution becomes less predictable. And it is this less predictability that is going to define the complications associated with understanding evolutionary processes. So, in the next video, we will start our discussion of three experimental fitness landscapes that have been made, and these three studies are all done in E. coli. The first one is a paper by Daniel Hartel's group in 2006 in the journal Science. The second one is a paper by Tim Cooper's group in 2013, also in Science. And the third one has to do with is a paper from Andreas Wagner's group. This was published late last year, also in Science. This will also give you a sense of, so there are papers that describe fitness landscape in E. coli of different scales from different cellular contexts, but they will also tell you that in the 17 years between these three experiments, how much progress have we made towards understanding and towards experimentally being able to decipher what are these structures called fitness landscapes. So, this is what we will start with in the next lecture.